Hello everyone, myself Abhishek Kumar from Group 316. My topic is active membrane transport. Active membrane transport plays a crucial role in maintaining the concentration of ions inside cells, such as high concentration of Na plus ions. This transport process occurs against the electromechanical potential gradients, and it's essential for the exchange of matter and energy with the surrounding medium. Active transport involve coupling diffusion fluxes to exerogenic energy from ATP hydrolysis. The coupling of chemical routine reaction to diffusion can be indirect through maintaining a stationary state or direct due to membrane anisotropy. Facilitated transport, a type of active transport, involves the presence of carriers in the membrane that interact with transported ions or molecules. These carriers form a complex with transported ions or molecules. These carriers form a complex with transported substance on one side of membrane, which is the broken down on the other side. The sodium pump is an example of facilitated transport utilizing the free energy of ATP to transport Na plus and Ka plus ions against their concentration gradients. This process involves the enzymes Ka plus and Na plus, activated ATPase and phosphorylate carriers that undergo dephosphorylation through conformational changes in virus regions of the membrane. Overall, active membrane transport is essential for maintaining cellular and enabling the efficient movement of ions and molecules across membrane. It allows cells to control internal environment, facilitate cellular process, and respond to external signals effectively. The mechanism involved in active transport, such as the sodium pump, highlight the intricate interplay between chemical reaction, energy utilization, and membrane dynamics in cellular functions of cell membrane. Active transport requires energy ATP since it takes molecules from a lower to a higher concentration against its concentration or electrochemical gradients. Importantly, active transport is necessary for homeostasis of ions and molecules, and a significant portion of the available energy goes towards maintaining the process. In particular, the sodium-potassium pump is required to maintain cell potentials. Secondary action potential can be seen in neural action potentials, where a hydrogen electrochemical gradient is established to synthesize ATP. An example of an antiporter is the sodium-calcium antiporter that exists in myocytes to maintain a low intracellular calcium concentration. And an example of symporter is the sodium-dependent glucose cotransporter that transport glucogalactose with two sodium ions into the cell. The mechanism of membrane transport. An example of primary active transport, the sodium-potassium pump directly utilizes ATP to bring three sodium ions out of cell and two potassiums pump directly via cycle of change to the shape of the protein pump. The protein is initially open to the cell interior, allowing sodium ions to adhere to high affinity pump. Second, binding of the sodium induces the phosphorylation of the pump via ATP hydrolysis. The shape changes also creates a high affinity environment to potassiums of ions can thus bind, causing the release of attached phosphate group. Removal of phosphate group returns the pump to its starting conformation, example, facing the cells inside. The establishment of an electrochemical gradient following this process mainly occurs via potassium efflux channels that allow the diffusion of potassium along its concentration gradients, such as electrochemical gradients, can then serve to power secondary active transport. Secondary active transport employs contrasporters transport multiple solutes, and they can be divided based on whether the transport used are symptoms or antiporters. Example, transporting solutes in the same or different directions. The antiporter utilize the energetically favorable movement of one solute down its gradient to allow the otherwise energetic energetically unfavorable movement of another solute against its gradients. The sodium calcium exchanger, for example, transporter and sodium ions into the cell in exchange for one calcium, out accomplished because of the previously established sodium concentration gradients.